Hello and welcome to Thoughts on Rice, a podcast hosted by the University of California Cooperative Extension Rice Advisors. I'm one of your hosts, Sara Marsh Janish, and I'm a rice farm advisor for Colusa and Yolo counties.
I'm Whitney from DeForest. I'm the Cooperative Extension Rice Advisor for Sutter, Yuba, Placer, and Sacramento counties.
My name is Luis Espino. I'm the Rice Farming Systems Advisor for Butte and Glenn counties.
I'm Michelle Leinfelder-Miles. I'm a farm advisor in the Delta region. I work on all sorts of field crops, grains and forages, but one of those is rice. And the counties that I cover are San Joaquin, Sacramento, Yolo, Solano, and Contra Costa counties.
Together, the UCCE Rice Farm Advisors seek to provide relevant, topical, research-backed information relating to California rice production. On this episode, we welcome back our colleague, Dr. Sara Rosenberg, a UCANR Farm Advisor for Mariposa, Merced, and Stanislaus counties. If you've been listening to the podcast for a while, you may remember Sara from last year, when we had her on to talk about her research into cover crops and rice systems. Now, Sara has recently published a paper as part of her dissertation work, the first study to assess soil health and agronomic outcomes in diversified and continuous rice production systems in California. This journal article, if you're interested in reading it for yourself, is called Benefits and Tradeoffs of Diversifying Rice-Based Cropping Systems, Impact on Soil Health, Productivity, and Agroecosystem Multifunctionality, and was published in Volume 391 of Agriculture, Ecosystems, and Environment. Although there is a lot to work with this paper, in this episode, we're going to focus on one aspect of this fascinating work: how crop rotation in rice systems affects soil microbial communities. So with that, I'd just like to officially welcome back Sara. So nice to see you. How are you?
Hi, Sara. Yeah, thanks so much for having me back on this podcast. I'm doing well, thanks.
Great. Sarah's are reunited. I love it. Now, I just want to check in with you. When we last talked, you had just begun in your position as a UCANR advisor in the counties of Mariposa, Merced, and Stanislaus. Can you give us an update on what you're working on or any exciting things that have happened over the last year?
Certainly, yeah. Well, I'd say that moving into year 1.5 of this new and very broad position, it's been quite a roller coaster. Up in the foothills, we've had a lot of really interesting kind of events and opportunities in terms of working with locals and local farms and local communities to lift up local agriculture through this really big annual event called the Agri-Nature Tour. So we did that just last month, and it was a big event where Mariposans and people that are coming into the area, mostly tourists into the Yosemite area, are touring local farms and learning about the local ag in the community and what's going on. Outside of that, we've been working with local vineyards and small farms to adopt cover crops and kind of look different cover crop species implementation, and everyone's been really interested in those things thus far. And yeah, I guess down in the valley, I don't know, things are a little slow moving, but more gearing up towards implementing some regenerative research in tree crops. So looking at integrated livestock management in pistachios and almonds. We've got a partnership going on at UC Merced's Smart Farm with the Almond Board and Cooperative Extension to implement a regenerative almond orchard demonstration. So kind of pushing the program towards the, cropping system design of regenerative orchards in the San Joaquin Valley. So those are a few highlights, I would say.
That's quite a bit. And we were talking about this a little bit earlier, but there's so much diversity and variability in the work that you do, but not only the work, but also the stakeholders as well. Would you say that it's an exciting challenge to try and bridge that gap between, like you said, the tourists who might be in the area for Yosemite versus the people who are landowners and managers?
Yeah, I mean this position and like all farm advisor positions, I feel like we just wear so many hats and we work with so many people. Like trying to narrow down the clientele is really just like it's based on your scope of work, but it's also a personal choice of like who you want to work with. And I think as the agriculture landscape and challenges change, so do our clientele. So for myself, really trying to lift up agrotourism in the regions of the foothills is going to mean working outside of the community clientele of farmers, but also in the Sierra foothills, landowners, people who are not quite defining themselves as for-profit farmers, but are really just trying to live off the land in a sustainable means. Those are part of my clientele too. And so kind of redefining the definition of a farmer up here versus what a medium scale commodity crop farmer looks like in the San Joaquin Valley.
That is really exciting. And I'm sorry to kind of stay on this, but this is so interesting because I feel like that might be a gap that we as an organization have kind of overlooked, that intermediary between production farmers and then those people who, as you said, are living off the land.
Oh, well, certainly. I mean, absolutely. I mean, in general, I think that parity wise, right, we just don't have the same population demand in the foothills as we do in the valley as far as like sizable operations are concerned. But I do see a shift, especially a lot of beginning farmers trying to, like, post COVID, moving into more of these rural forested parts of California. And a lot of them don't know what to do, where to start. They have this kind of idea where they want to, lower their footprint. They're also moving into these regions that are really, really high risk for things like wildfire, right? And so there's a nexus of, as I've been up here and learning about these things, of how do I maximize sustainability, biodiversity, habitat, conservation, but also fire mitigation. And where does that come into play? So a lot of really interesting collaborations with different folks up here. The Fire Advisor, for one, the Master Gardeners program, people like that.
We do love a collaboration, and it sounds like you've really hit the ground running and started some very interesting work. Later on in this conversation, I'll actually ask you to share some links and information so that we can direct people in the area who might be interested. With that, although it's a bit of a shift from your usual day to day, I do want to kind of talk about something you used to spend quite a bit of time on when you were up here, up in the north with us. And that's kind of about this project that you worked on a few years ago as part of your dissertation. And so, first off, congratulations on getting this journal article published, Benefits and Tradeoffs of Diversifying Rice-Based Cropping Systems, Impacts on Soil Health, Productivity, and Agroecosystem Multifunctionality.
It's quite a mouthful, I know.
Well, it is, but you've got so much to cover in this article, and I'm just going to caveat this. I've read the article a few times, definitely not an expert, but it's really interesting how much you all managed to combine and analyze. Can you just, I guess, briefly, for everybody, kind of give us an explanation of the rationale behind this study?
Sure. Well, I've always been interested in multifunctionality as a concept in agriculture, which is really about like laying out the implications of an agriculture system across many dimensions. I think it's pretty traditional that we have the lens of success of agriculture coming from one or two different indicators, right? Whether that be yield, whether that be economics, profit, some type of productivity indicator, right? But multifunctionality is the concept that says agriculture landscapes can provide multiple services beyond producing food and fiber. This looks at biodiversity conservation, the contributions to water conservation and water cycling events, the contributions to different socioeconomic viability outcomes, and even perhaps cultural outcomes. There's many different multifunctionality frameworks that one can use, but I really wanted to take that framework and say, how do rice systems that are under this, diversification practice of crop rotation and then continuous rice systems differ across all of these different dimensions? So that's why it's kind of like, analyzing so many different aspects rather than just one or two things.
Absolutely. And I think part of the reason you all looked at this is because of the way that multifunctionality can affect resilience. Is that right? Can you define for us what resilience means here?
That's a great question. I mean, I would suggest resilience means the ability to withstand shock, right? In terms of an ecological definition, it looks at how a community or a system recovers after some type of turbulence or some type of shock event. And usually what we see in an ecological systems is that the more ecosystem services that are functioning in that system, the more resilient it's going to be, or the more it's going to be able to recover quickly after a shock. And so if you just kind of take that theory and say, well, if you have more ecosystem services available on an agricultural landscape, then you could correlate that to a more resilient system. And I think that was kind of the rationale behind it. Taking a step back a little bit more, I think originally we were looking at this really more from a reductionist lens where we were thinking, well, let's look at this from a weeds standpoint or from a soil health standpoint or just one or two of these things. And as I, was kind of building in my PhD world, grad school, I said, I don't want to just take that reductionist standpoint. I'm interested in the full on picture of the trade-offs across all these different dimensions, looking at agronomic, looking at environmental, looking at soil health, many different things. So, yeah.
You did mention the trade-offs. Can you tell us some of the trade-offs that can be expected when increasing the number of functions within a system?
Certainly. So generally, the more ecosystem services you can provide, the more resilient you are, but the less likely you are to maximize any one service, right? And so that's the trade-off. So if you are looking at maximizing one service, like let's take nitrogen availability with the hopes of maximizing your yield, whereas nitrogen availability is a supporting service and yield is considered a provisioning service, you are going to likely see a reduction in other services. For example, you may sacrifice carbon sequestration because to sequester carbon, you need to immobilize nitrogen and microbial biomass. Or you may lose carbon in other ways in order to increase nitrogen availability. You may lose certain biodiversity services because you are shifting communities that are highly adapted to nitrogen-rich environments. Or you may have trade-offs with higher pest populations, et cetera. So this is what, current literature tells us. But at the same time, the more ecosystem services you can enhance on the farm, the less trade-offs you have between them.
Thank you. That was a really good way of tying that all together, by the way.
Okay, good. I know these things can be a little bit, I don't know what the word is, nuanced or theoretical, I guess, sometimes, and trying to tie it to the applicable and is difficult.
Mm-hmm. But I mean, we're talking about a pretty complex subject as a whole. How did you and your team design this experiment to study this concept? What was your, I guess, basic design and methods for undertaking this project?
Yeah, so this was an on-farm field experiment, meaning I sampled across like many different farms that had been under different management regimes for long term. So every farm was sampled from once in order to capture what we call the legacy effect of that management regime over time. And that's as like in comparison to a trial that might be replicated on a research site, and you're doing those research trials year after year after year to assess change. We just sampled from a large number of farms in the Sacramento Valley. Each farm was sampled from once for soil health. We got survey data from growers for management, and then we just assessed that those farms that were continuously cropped with rice compared to those farms that were under some form of crop rotation. delineated from conventional versus organic.
When you talk about you've sampled in the Sac Valley, you probably were able to encompass a wide variety of soil types. Did you see a lot of change in the sampling sites?
Yeah, I mean, in general, that's going to be, a control issue. And most rice in the region, as you know, they're going to have pretty high clay content, pretty much in the clay or silty clay loam texture. They're also going to have attributes like soil that's poorly drained due to floodplains or impervious clay pans or hard pans, things like that. So what we do to try and control for that variability is if we had like 46 sites that we sampled from, we tried to get comparisons of the rotated field in proximity to the continuous rice field, geographically speaking, with similar soil types. Sometimes that was difficult to find, which is why you get more, you can just get more field samples that kind of overshadow that variability in the analysis. The other part about this is that the majority of rotations are happening in certain regions, right? And so if you look at the field selection on a map that we had, most of the rotated fields were down south, which will also skew certain things. So we had a high density of sites that we sampled from in the Sutter, Yolo, Calusa region. And that's just a product of where rotations are happening in Sacramento Valley.
In a way, it's more realistic, at least.
Yeah, absolutely.
And so how many years, I forgot to ask this earlier, but how many years did you collect data for this project?
It was over 3 years. But again, every site was sampled once. So we didn't go back to those farms and sample, consecutively every year. It was just about like finding new sites every year for 3 years. And we sampled between 10 and 22 fields per year.
Mm-hmm. And I guess in terms of the fields that you were sampling, I'm interested in some of the management practices used in these fields just to kind of set up. Are there records of some of your rice farmers doing winter practices like winter flooding? Or is there a certain percentage of organic or conventional fields that you were able to get a history of?
Yeah. So I guess the first question is management practice around the winter flooding, which, as we know, is extremely common in most of the Sacramento Valley for continuous rice farmers. And so the majority of those who were continuously cropping rice also did winter flooding. Those who were doing rotations were not doing a winter flood because it would impact their ability to come in the summer and do an upland crop. That wasn't rice for a lot of different reasons. But oftentimes, those farmers, and this gets out of our time sampling horizon of when we were looking at some of the soil health stuff, but oftentimes the growers that were considered like they were in the category of like rotating rice with summer crops, when they were in their rice system, they would be in that system for anywhere for like 2 to 5 years, sometimes more. And during that time frame, they would winter flood if it was once it was rice back to back to back. So, not clear cut. Yes, they did. No, they did not. But during the rotation phase, they did not winter flood. And then what your question about like organic versus conventional numbers, I think. That was it? The majority of the sites were conventional. 34 were conventional and 12 were organic. And so that's a disparity. That is something that was recognized throughout the study where we said anything, that we're able to pull from this as like an organic effect is a trend because the population size was so small. Of the 12, 8 of the organic fields were under some form of rotation, and 4 of them were continuous rice practice. And the organic rotated fields they were the ones that were the most diversified outside of rotation practice, meaning they also implemented a lot of cover cropping, soil amendments, chicken manure mostly, sometimes compost, I believe, but most, or if not all of them, did some form of organic amendment and cover cropping regime along with that rotation.
I wanted to ask how you were taking your soil samples in each of these fields, just kind of the methodology for that, and then how you analyze them later.
So the methods for sampling was quite fun, actually. We were able to get into the fields in spring, right after first or second tillage pass, and before any inputs were put in place. So that'd be between March and May. And I would be out there with myself or a team or a few people, and we'd, I'd be driving around and kind of scout the field and take a representation number of soil samples from each farm. So anywhere from 10 to 22 plus soil samples were taken. These are sites that are 30 acres to 150 acres that you're driving around on, pretty rough soil that's been tilled up, and it's kind of like driving on a moonscape sometimes, felt like. But yeah, we'd sample down about 6 inches, just down to the annual cropping system root level recommendation, and aggregate everything, mix it all up together. And then we put soil samples in storage based on how they needed to be preserved for the type of sampling that we were doing. I sent out all of my samples to labs, thankfully, so I didn't have to do the in-house work of all of that analysis. I was more focused on the in-field sampling and getting the survey results from the farmers and then doing the data crunching on the other end.
And one of those analysis methods that you talked about was phospholipid fatty acid, or PLFA. And that's not one that I'm terribly familiar with. Can you explain how you use that analysis to provide information on microbial biomass diversity?
Yeah, sure. So I wanted to look at the microbial biomass differences between the sites, but I also wanted to look at the diversification of the different groups of microbes across each site. And PLFA is one way of doing this. It's going to give you the total living microbial biomass, whereas a lot of times a soil analysis on microbial biomass is necromass and living. Both necromass and living are good in things to know about, but this one is going to look particularly the living microbial biomass. And that's because of the way that it extracts. I'm going to get kind of in the weeds here as far as I know, which isn't as much as some, but basically it's extracting certain lipid chains from the microbial biomass and then analyzing them to look at the different fingerprints that are going to change across the groups. So those types of lipids, they degrade quickly. And so after microbial lifespan is overturning in the soil, those lipids aren't going to be able to be sampled from anymore. So those kind of fingerprints reveal differences across the different phyla groups of microbes. Those would be things like different types of bacteria, different types of fungus that is in the soil. I mean, those are the two big groups, right? And then within that, giving you the fungal to bacteria ratio, and there's just like a lot of information that you can get. So it's a breakdown of who's in my soil rather than just how much is in my soil.
That is a really, really neat. Before you got the results back, did you have any kind of hypothesis or idea of what type of microbes you were expecting to see?
I mean, as far as like hypothesis is concerned, we kind of went based on literature that said the more diversity you have above ground, the more diversity you have below ground type of logic. But generally, I was like going into this more of an unknown exploratory lens because it's not apples to apples. You're looking at a system that is semi-aquatic. The biogeochemistry is completely different under standing water for many, many of those months versus an aerobic cropping system that is dominated in oxygen. The amount of biomass is different across the systems when you're talking about, turning over the last year's crop. And all of that's going to affect who's eating what and when and how. So I was just kind of more of taking that exploratory view of what's the difference between these two systems.
I mean, I think that's really neat, though, because to keep it simpler, you kept an open mind as you were going into it. You didn't have any preconceived ideas. And part of that is because, like we said earlier, this is kind of a landmark study. This is the first study to assess soil health and agronomic outcomes in these kinds of systems. So not a lot to go off of in terms of literature.
Yeah, well, I think also, to be fair, not a lot to go off of in the context of California. There's a lot of literature that has been studying crop rotations with rice in subtropical environments, in the kind of paddy environments up in parts of Asia that have 2 seasons or, but those systems are so different compared to what we have going on here in our rice realm. So yeah, there was a lot of unknowns going into this paper in the context of California agroecosystems.
So I did notice in your paper, you noted there were higher carbon and nitrogen mineralization rates in rotated rice fields. And you mentioned that the differences in the bacterial communities between these systems may have contributed to the shift. Can you walk us through that statement?
Certainly. Just a little kind of like breakdown of what we found along the soil microbial differences. Generally speaking, there were 2 groups that differed within the rotated fields and the continuous rice fields. And it wasn't that there was more diversity between one type or the other. It was more that there was a shift in the communities that existed, which really starts to make sense when you think about it, because you're changing the diet, of these microbes pretty vastly when you move from a rotated system to a continuous rice system. So in the rotated fields, which were dominantly conventionally rotated fields, we saw significantly higher rate of a group of bacteria called actinomycetes. And these are beneficial bacteria that promote soil health as secondary decomposers of organic material. They are really known for their role in cycling nutrients and mineralization, particularly of secondary decomposition of things like cellulose, hemicellulose, lignin. And they are also known, for it's a very, very large phyla group. So getting into the nitty gritty of what types were there, we didn't get that far. But this group is also known for suppressing plant pathogens. Actinomycetes are previously known as the actinobacteria, or classes of bacteria that we know are in our kind of world as like antibiotics, so to speak. So they're the good bugs in some way. So that's what that kind of like correlation to higher carbon and nitrogen mineralization, because we did see a reduction in POC, which is kind of an indicator for that labile carbon in that easily degradable carbon bank. We saw a reduction of that in our rotated fields. We also saw a high reduction of our ACE protein, which is the indicator of our readily available organic pool of nitrogen in our rotated fields. And so we saw the correlation between those reductions of those carbon and nitrogen pools and the higher amounts of actinomycetes as, perhaps these types of bacteria are driving that reduction or that mineralization of carbon and nitrogen.
And that does make sense, given what you've said about what that population, what those can do and how they act within the soil, depending on the various types of food that they're getting into their diets.
Exactly.
Now, When you're looking at the soil microbial communities, was there a noted difference between the different management methods? So, say a difference between the organic rotated fields, the conventional rotated fields, and the conventional non rotated fields?
Yes, and yes, and.
That's a great answer.
Or yes, but I'm not sure. I like yes, and. So I mean, you did see it. And as far as the like what we'd call the significance statistically speaking, it was less of a significance, and that is because honestly, we just, like I said, didn't have enough organic fields. Right, we had a very small amount of organic fields. But you do see a trend in organic rotated fields and organic non-rotated fields to have higher amounts of fungi present compared to the conventional fields. And this is a particular type of fungus that is the saprophytic fungus, which is another group, a phylogroup that is recorded in the phospholipid fatty acid analysis, the PFLA. And this also, the sort of higher saprophytic population in our organic fields compared to our conventional fields makes a lot of sense when you think about it, because this population is really sensitive to things like herbicide applications. It's really sensitive to pH change. These are primary decomposers. These are our funguses that really help break down our organic material at the higher level. And so when you think about all of the different organic matter amendments that organic farmers are using, like chicken manure and cover crops, this is going to be a big kind of like feast, so to speak, for our saprophytic groups of fungus. There's also some evidence around these particular primary decomposers being really good at breaking down lignin. This is our like really woody type of material. And even thinking about on the organic continuous rice fields and on the conventional continuous rice fields, the amount of rice straw that's going into that input, right, is going to need a starting point to break down before the actinomycetes can come in and break it down further. And so those funguses, those saprophytic communities are going to be the primary stage colonizers at that amount of biomass compared to like a conventional rotated field that's going to just have less of that type of biomass circulating in the system.
And so because maybe a conventional continuous field might have exposure to more chemicals that may hinder the population of the saprophytic fungi, that might indicate why there were higher populations of those in the organic fields. Is that accurate to say?
Correct. Right. So we looked at these results in terms of getting significant indicators across population sizes. We put them through what's called a multifunctionality analysis and lens and a multivariable assessment. And we saw correlations between these two. And then, we can interpret some of these findings through our understanding of the mechanisms behind them.
Yes, very, very much so. I'm really glad you keep mentioning that, because oftentimes research like this does get distilled down a little bit too much, but there's a lot of variables at play here. And I guess one of those interesting variables was the difference between rotated and non-rotated fields in terms of total microbial biomass. So not just microbial diversity, but was there a difference in numbers of total microbial biomass between these rotated and non-rotated rice fields?
We expected there to be, but there wasn't. There wasn't a significant difference between the two. So, like I said, it wasn't like a complete difference in terms of diversity. It was just a shift in community. And when you change the community, you're not necessarily changing the entire biomass of microbes. So that, again, is at a time point sensitive sampling event. So we don't know how things might change later in the season or at a different time frame. But the time that we sampled in spring, when rotations are coming, are and I did not mention this in the methodology of the study, but at this time that we sampled, all farmers that were rotating with rice were going to be going into a rice crop the following year, coming out of a rotation for 3 to 5 years or whatever. I think actually it was, some of them were even 1 to 6 years or something. So at that time point when we were sampling, we did not see any differences in total microbial biomass.
And so that could be different, I guess, than what people might expect. Some may think that a flooded rice soil may be, because of the anaerobic environment, having less of a microbial biomass. But can you say why the flooded rice soils might contribute actually to microbial biomass?
Yeah, and it was kind of a toying, like, as far as, literature is concerned, you could have gone either way. You could have suggested like there would be more microbial biomass in the rotated fields. There's a lot of really good studies out there that actually just show how efficient paddy soils and rice systems are at building carbon reserves, and this is directly correlated to microbial biomass. And so in general, you could expect a higher microbial biomass in the rice fields. I do think that is still prevalent in California systems, right? Like that we are in doing the rice systems in California are a major carbon contributor in general, net carbon contributor, because of the high biomass turnover of rice stock and the flooded environment. And the flooded environment slows down decomposition. So those two kind of mechanisms are going to differ from anaerobic system, where you are rotating with annual crops that may not have as much biomass turnover, and perhaps like the type of biomass is not as difficult to decompose like rice straw is. So you may not have as much biomass, but you're also under an anaerobic environment, which is very inefficient. And so you have a lot of CO2 respiration happening and loss of carbon through that CO2. So I do think that is, that's knowledge that we knew, and that's something that this study backs up. But there was kind of this other side of the coin, which is that, well, then why did the rotated fields not show a drop in overall microbial biomass? And, there's some interpretation there, but there's not a whole lot of causation. But generally speaking, the sites all had a pretty high level of carbon to begin with. California agriculture is intensive. And so, even in the annual cropping system, you're still getting a pretty good turnover of organic material after the season. And that kind of high amount of organic matter in the soil across the sites just could not have any substantial decrease over time based on how stable the carbon levels are in the soils.
It's a complicated topic, and there's a lot of different factors, but it's so interesting just to see the output of this work, and especially when you're able to kind of illustrate it through these studies into microbes and the microbial communities. Now, one more thing, and this is a bit more rice related, but with the talk about how the straw takes a long time to break down and how that can affect depending on the different management situations, let's talk about straw incorporation effects. Anything that we can extrapolate from this study on how straw incorporation into the soil might have an effect on microbe communities?
The simple answer is yes. Rice straw incorporation can affect microbial communities. As far as the implications of that and what that means, I think the study does suggest a strength of the rotations is that you had more efficient nutrient cycling events happening. Mineralization was occurring at a higher rate in the rotated fields compared to the continuous rice fields. And so, if you wanted to, we also did see a pretty distinct increase in rice yields coming out of rotation. And that could be an effect of the weed support that the rotations were having, or it could be an effect of the nutrient cycling support that the microbes were providing, probably a combination of both. I think that it's hard to, it's this project brought up more questions than solid answers, as any good research will do. And for me, a part of that was, well, what would happen later on in the time series in the rotation sequence? So at the time of coming out of rotation, we saw this shift in microbes, right? Actinomycetes are really helping with cycling your nutrient level and breaking things down, but you're also getting a dearth in your labile carbon pools and your retention of nitrogen overall because you're cycling it through. But what happens like 3 to 5 years down the line when the rotation has been in rice for 3 to 5 years? Do those pools recover in some way? Is there this kind of like push-pull lever effect that happens in a rotated field in terms of nutrient cycling that may be a little bit more sustainable than just saying, oh, you're going to have a reduction of carbon and a reduction of nitrogen compared to your continuous rice fields? Those things weren't like, those were kind of questions that came up out of this. Generally, continuous rice fields are going to have some immobilization issues. And there's a lot of work from, I believe, Bruce Lindquist's lab that's talked about this. And so the rotated fields might perform better because it doesn't have those immobilization issues. But as far as climate mitigation is concerned, if you just take from the standpoint of when we sampled from, you're losing that carbon bank, which is affecting a lot of different things. So I know that wasn't like a direct answer to like, how does rice straw affect microbes? But it's just like the microbes are an indicator of so many other things, And so it's more like, how does the rice straw affect all these other things? I hope that was kind of a good way to think about that.
Absolutely, absolutely. And it's not just that we're interested in the microbes for the microbes' sake, but they are, I guess, an indicator of all of that's going on underneath the soil surface. And so it's been such a treat to read this, honestly. Actually, I copied this down because I was really interested in it. It's one of your final remarks on the results of the study, and I'm going to quote this. While soil microbial biomass was similar among cropping systems, microbial communities under diversified rotations trended towards higher bacterial abundance associated with mineralization processes. And I feel like that really sums up what we've talked about here today. I think that's probably one of the coolest things I've read coming out of this paper. And I don't want to take all the time to talk about it. This is your project, but anything here you want to add on this?
Well, I think that was a very concise way of saying what I was, trying to articulate. And yeah, I do think that was a really cool finding that came out of this project. There's so much more depth that can happen in any number of the dimensions that were analyzed in this paper. And that's one of the strengths of this paper is there's a breadth of knowledge here. And then one of the weaknesses is that it is, it's a breadth, not a depth. So, so much more work that needs to be done, I would say.
Well, and since you're in extension yourself, I am going to ask you, these results, how would you say they might apply to growers or producers or people who are interested in crop rotation?
Yeah, I mean, I would suggest, looking at this as a body of knowledge that can display benefits and trade-offs. It's not like a best practice approach. It's not like do the study and then say best management practices, therefore are to X, Y, and Z. This is more of a displaying all of the benefits of this type of system and the relevant trade-offs of this type of system and all of the benefits of continuous rice systems and some of the trade-offs of that system. And then as a grower, you can be informed and make decisions based on your own personal sustainability goals of what you might want to do to, improve upon sustainability outcomes. I understand and knew growing into this project that rotations for rice growers in Sacramento it's not something that's going to become common, and it's not a super feasible practice. But, in our changing climate and in our changing environments, having a tool belt that allows you to adapt is really important. And rotations are just one of them among many.
Well said. Well said. With that, Sara, I just want to ask you, well, first, thank you for your time and ask you if there's anything you'd like to share concerning your program or your work here with our listeners.
Well, if you want to follow my program easily without much need for deep diving and reading right now, you can follow us on Instagram and Facebook at UCCE Regenerative Agriculture. And that's going to give you some good highlights into the different projects that's going on. I'm really excited to see the research unfold in some of the arenas of integrative livestock management and tree crops in the San Joaquin Valley. And while I don't have anything firm to share with you, following us on Facebook is definitely a good way to see how that carries out. We do have a lot of really fun events up in the foothills in Mariposa. If anyone is interested in learning more about the type of ag that's going on up here and the local food systems that are growing out in this region as well. So yeah, that's the best I can give you right now. UCCE Regenerative Ag. Follow us on Instagram and Facebook.
Sara's being modest, but absolutely, I've been watching their work there, and especially the venture that they put forth about a month ago with the agritourism. That was really impressive to see. So please stay tuned, go check out that. And if you're in that area, if you're touring around Yosemite, come on by. Man. Well, Sara, thank you so much for coming back on. I know this is no longer technically in your program area, but we really appreciate you taking the time to talk about the time you spent with us up here in the North region. So it's really a treat to see you again.
Thanks, Sara. And I do have one more small announcement that I totally blanked on, which is the third aspect of this dissertation work will be published quite soon. And that is an entire paper on the economics of crop rotations with rice systems. So that's the third lens of this project, right? And that looks at comparing a rotation over a period of 15 years to continuous rice and looking at profitability outcomes. So stay tuned for that, I suppose.
Yeah, we're definitely going to please beg you to come back on to talk about that when that comes out. We're really excited about that one. Thank you so much for your time, and we hope to see you again soon.
All right. Thanks, Sara. Take care.
With the new year, we've got some new events we'd love to share with you all. First up, we will be holding our UCCE Rice Winter Grower Meetings. Those will take place from January 21st through January 23rd. As always, we do a series of 5 meetings. 1 in the morning, 1 in the afternoon, and the final day will just have 1 in the morning. The Butte County meeting will take place January 21st, 2026, which is a Wednesday at 8 a.m. at the Evangelical Church in Richvale. The Willows meeting will take place the same day, January 21st, 2026, at 1 p.m. in the Willows Memorial Hall in Willows, California. Please note this is a new location for us. The Colusa meeting will take place January 22nd, 2026, which is a Thursday, at 8 a.m. in the Colusa Industrial Park Conference Room in Colusa. The Yuba City meeting will take place January 22nd, 2026, which is a Thursday again, at 1 p.m. in the Veterans Hall. The final meeting will be the Woodland meeting on January 23rd, which is a Friday, 8 a.m. in the Yolo County Farm Bureau in Woodland. We will also be holding a rice production workshop this year. As a reminder, this is a meeting that was postponed from last year, and the date of that meeting is March 18th and March 19th, as this is a 2-day meeting. This is a meeting which is an in-depth workshop that covers the principles and practices of rice production in California, best suited for beginners or newcomers to the California rice industry. Our last save the date is the Rice Quality Workshop, which will take place July 30th, 2026. As a reminder, if you're interested in learning more about any of these events or save the dates, they will all be posted on our UC Rice website, which is once again agronomy-rice.ucdavis.edu. For more information about these and other upcoming events, feel free to check out our resources, which include the UC Rice blog and the UC Agronomy Rice website. In terms of other resources you might want to take advantage of, you can also look at our newsletters, which include Rice Briefs, which covers close to Yolo, Rice Notes, which covers Yuba Sutter, Rice Leaf, which covers Butte and Glen, and Field Notes, which covers rice in the Delta region of California. Thanks for listening to Thoughts on Rice, a University of California Cooperative Extension podcast from the University of California Agriculture and Natural Resources. You can find out more about this podcast on our website, thoughtsonrice.buzzsprout.com. We'd love to hear from you, whether it's from using our text link in the show notes, a survey submission in our feedback form, also in the show notes, or in a comment or rating on your podcast streaming service of choice. We are excited to announce that after several months of trying, we are finally on YouTube. So if that is your podcast streaming service of choice, you can now listen to us on YouTube. You can also email us with any comments, questions, or concerns at smarsh@ucanr.edu. Rice harvest is finished. Fields are flooding up, and the migratory waterfowl are beginning to arrive. Remember, like the growers like to say, have a rice life. Mention of an agrochemical does not constitute a recommendation, merely the sharing of research findings. Always follow the label. The label is the law. Find out more at ipm.ucanr.edu. The views, thoughts, and opinions expressed are the speaker's own, and do not represent the views, thoughts, and opinions of the University of California. The material and information presented here is for general purposes only. The University of California name in all forms and abbreviations are the property of its owner, and its use does not imply endorsement of or opposition to any specific organization, product, or service.